Alright guys, here we are with concept 23.2. It says the Hardy-Weinberg equation can be used to test whether a population is evolving. And that word equation hopefully is not freaking you out there. Surely you didn't think you'd get through biology without using your calculator and pen and pencil and mathematical skills, did you? So a little bit of math here. We'll kind of step through it and make it as painless as possible. And here it says the first step in testing whether evolution is occurring in a population is to clarify what we mean by population. Always helps to understand our terminology. So let's first define what is meant by that word population. A population is a localized group of individuals capable of interbreeding and producing fertile offspring. So a population we're going to find can be geographically separated from other populations, or it can be separated from other populations by habit, or a variety of other things can do this. But so a population is a distinct group, a localized group, so they're going to be kind of within the same area of creatures of a particular species that interbreed and produce fertile offspring. This population is going to have what we call a gene pool, and the gene pool consists of all the alleles for all the loci in a population. Whereas last semester when we talked about genetics, we kind of talked about just say two parents and a Punnett square, and we were crossing those two parents and we were only looking at that particular cross. Well, in this particular chapter when we talk about genetics, we are pretty much going to dump all of the alleles in one big bucket. And so since we are talking about the entire population of this creature, of these creatures, we're not talking about any one particular mating pair. We're going to take all the alleles and just dump them into a bucket, really regardless of who those alleles came from or any one particular creature. So it says a gene pool consists of all the alleles for all the loci in a population. A locus is fixed if all individuals in a population are homozygous for the same allele. So for example, if there's a population of deer and I don't know, let's say there's a trait for how much hair they have on the tip of their ears, and every deer in that population is homozygous for that trait, for that particular locus, well then you would call that fixed. It's not going to change. If everyone has the exact same allele at that particular location, well then that is a fixed trait or a fixed locus, and we know we're not going to see any change there because we don't have any variety, no variation. In other words, there can be no variation because there's only one possibility. So that would be called fixed. So let's take a look at a quick little map or a chart that you can see in your textbook. This is figure 23.6, and it shows two populations of caribou up in the Yukon, kind of in the Alaska/Canada area here. They're not totally isolated from each other. You do see that the ranges of these two populations pretty much overlap, but that doesn't mean they're all together at the same time. Caribou tend to range as the season changes and they migrate from one place to another, so at times they may overlap, but generally speaking we're saying that this group, the pink porcupine herd group, tends to breed with each other, and the 40-mile herd range group, the bluish group, they tend to breed with each other. So we would still consider them two separate or distinct populations. So again, remember that we're talking about just putting all the alleles in a bucket, and the allele is, say, capital A and little a for a particular trait. If we're only talking about there's only two possibilities there. So the frequency or the proportion of any one particular allele in a population can be calculated, and that's where some of our math is going to come in. For diploid organisms, which means they have two sets of chromosomes, like humans, we have 23 pairs, so we're diploid. For diploid organisms, the total number of alleles at a locus is the total number of individuals times 2. So if you have a population of, I don't know, let's say 10 caribou, and at each particular locus the caribou have two alleles because they got one from mom and one from dad, well, the total number of alleles at that particular location is the number of caribou 10 times 2 because they each have two alleles. So there would be 20 possible alleles at that locus. The total number of dominant alleles at a locus is 2 alleles, homozygous dominant. That makes sense. And then plus 1 allele for each heterozygous individual. And the same logic applies for the recessive alleles. So we could get a complete count of all the alleles. If you count all the homozygous dominant alleles, plus you get one dominant one for a heterozygous creature, and then you could count all of the homozygous recessive alleles. There would be 2 at each location, plus you get 1 for each heterozygous creature. You could get a complete count or a complete record of all the possible alleles, and then you could start to work out percentages in a population as to which allele was most common or which allele you saw most often in what combination. Just like in mathematics and chemistry, we like to use a lot of shorthand just to make things easier, and I can't tell you why they chose the letters P and Q, but that's just the way it is. It's by convention. So by convention, if there are 2 alleles at a locus, and that's all we're going to be really looking at for the most part, is a trait that has 2 possible alleles, a locus with 2 alleles, then P and Q are used to represent their frequencies. So the 2 possible alleles, their frequencies are going to be represented by the letters P and Q. I guess they chose P and Q because when you write them, they pretty much look opposite of each other. The frequency percentage-wise of all alleles in a population will always add up to 1. Of course, that is 100%. So you have to end up with 1 as your final answer, or 100%. So for example, P plus Q is going to equal 1. You've got to come out with 100%, or you know you've got your math wrong. So let's step through an example. We'll go slow here. Let's kind of go back to a population of wildflowers that is incompletely dominant for color. So you can have red flowers, and that would be if they are homozygous dominant. Then you can have white flowers, that would be if they are homozygous recessive. And then you could have pink flowers, because remember we have incomplete dominance here. That would be if they are heterozygous for these traits. And then let's say we count all of the flowers in a particular population, and we keep a record, and we find that there are 320 red flowers, there are 160 pink flowers, and 20 white flowers. So there are 320 flowers that are homozygous dominant for the trait, 160 pink flowers, we know those are heterozygous, and 20 white flowers, we know those are homozygous recessive. Now there's only 2 alleles. There's only either the R allele or the W allele. We can calculate the number of copies of each allele in this entire population. For the R dominant allele, it's 320 times 2, because of the 320 red flowers, they have 2 copies. And then plus 160, that's half of the heterozygous. So there's 800 copies of the R allele. And then of the W allele, we have 20 individuals that have 2 of them, so that would be 40. Plus the 160 from the heterozygous. So you have 200 copies of the W or the recessive allele. To calculate the percentage or the frequency of P, which is going to be our dominant R allele, we take the number of R alleles that we had, 800, divide that by the total number of alleles that there are, which was 1,000, and we get 0.8 or 80%. And the Q, we do the same way. We count the frequency of all of the W recessive alleles that we found. That was 200. Again, divide by the total number of alleles in the population, which was 1,000, and we get 0.2 or 20%. And the sum of the alleles, remember, has to equal to 1. 0.8 plus 0.2 does equal to 1. So we come out with 100%. So we know we have everything calculated properly. For loci that have more than 2 possible alleles, which we're not really going to look at, but it works the same way. If you're talking about a particular loci that has 3 or 4 alleles, well, the sum of the allele frequency still has to come out to equal 1 or 100%. So it works the same way, just a bit more complicated, a few more numbers to write down. Alright, so let's put some of these numbers to work. There is something we're going to look at called the Hardy-Weinberg principle, or the Hardy-Weinberg equation. The Hardy-Weinberg principle describes a population that is not evolving. So what we're saying is, if a population does not meet the criteria of the Hardy-Weinberg principle, it can be concluded that the population is evolving. So we're going to find there's basically like 5 criteria that we're going to look at. And if the population doesn't meet all of these criteria, then you can conclude that the population is changing over time and is evolving. So the Hardy-Weinberg principle describes a population that is not evolving. And you're going to see, if you study population genetics or ecology or biology of populations, you're going to see that almost all, I mean virtually all, the vast majority of natural populations do not meet all the criteria of the Hardy-Weinberg principle and are thus evolving. So think of this as a tool. Think in biology and ecology problems, saying something like, assume a population is at Hardy-Weinberg equilibrium. That's kind of the equivalent of when your physics teacher tells you to assume no frictional effects when you're working out a physics problem. It's pretty much a setup. It's not reality. So the Hardy-Weinberg equilibrium that we're going to look at, you pretty much never see it in nature. But it is a tool to compare to reality to see how much evolution is going on. So if you hear that assume a population is at Hardy-Weinberg equilibrium, just know that that's very similar to your physics teacher telling you to forget that there's friction in the world, because it makes the problem a lot easier and you're dealing with this sort of simplistic, perfect equation that gets a lot messier in real life. So why would we use this tool? Why would we use this Hardy-Weinberg equation that virtually never actually exists in nature of this non-evolving population? Well, one way to assess whether natural selection or other factors are causing evolution at a particular locus is to determine what the genetic makeup of a population would be if it were not evolving at that locus. And then we have something to compare to. We can then compare that scenario with data from the real world, and if there are no differences, we can say that, okay, there's no evolution going on. But if there are differences between the real world data and the Hardy-Weinberg principle or equation data, if there are differences, this suggests that the real population may be evolving, and then we can further investigate to try to figure out why, what is causing this shift in genetics or this change over time. The Hardy-Weinberg principle is named for a British mathematician and a German physician who actually respectively independently derived it in 1908. So we have this British mathematician, last name Hardy, and this German physician, last name Weinberg. They were working on this issue separately, apart from each other, but they actually came to the same conclusion and answer together in about 1908, about at the same time. The Hardy-Weinberg principle states that the frequency of alleles and genotypes in a population remain constant from generation to generation. If it does remain constant, we call that gene pool in Hardy-Weinberg equilibrium. Now, you're virtually never going to see this in reality, but remember, this is a tool for us to use. In a given population where gametes contribute to the next generation randomly, allele frequencies will not change. Now, that doesn't mean that genotypes don't change as far as how many homozygous dominants and heterozygous and that type of thing. But remember, we're looking at the individual alleles, the big R's and the little r's. Well, in a given population where it's completely random as to which gametes get passed on, allele frequency will not change if no evolution is occurring. Mendelian inheritance preserves genetic variation in a population. And again, recall that this means if no evolution is occurring, if this particular population is at what we call Hardy-Weinberg equilibrium. Here's a visual to kind of help you understand. And again, the major difference between what we're talking about now and what we talked about last semester was here, instead of considering the possible allele combinations from one cross, one set of parents, one particular mating when we were doing a Punnett square, here we're talking about the combination of alleles in all of the crosses in a population. We're taking all of the alleles of that population, just sticking them into one giant genetic bucket, and we're randomly pulling from that bucket. If the mating is truly random in that population, you're not going to get a change in allele frequency over time. That, again, is if we're at Hardy-Weinberg equilibrium and no evolution is occurring. So Hardy-Weinberg equilibrium describes the constant frequency of alleles in such a gene pool that's in that particular state. So go back to the wildflowers that we were talking about. For example, the same population of 500 wildflowers and the 1,000 alleles where the P frequency was 0.8, 80% of the dominant allele, and the Q frequency, the recessive allele, was 0.2 or 20%. We can use these numbers, which represent the frequency of the alleles, to then calculate the genotypes, the frequency of the genotypes. So let's see, for homozygous dominant, we're going to have 2 of the dominant alleles. So we're going to do some multiplication using our rules of multiplication. That would be p squared. So our 0.8 times 0.8, or squared, is 0.64. And then we're going to look at the heterozygous. That is represented by 2pq. So we have 2 possibilities. We could have PQ or we could have QP. and that's why we have the 2 there. And that's going to be 2 times 0.8 times 0.2. Calculate that out, it comes out to be 0.32. And then we have the homozygous recessive, which would be Q squared. And that is 0.2 squared, which would be 0.04. So now we can look at the frequency of the genotype, and we could confirm that with a Punnett square. So now we could say that 64%, or 0.64 of this population, will be homozygous dominant, 32%, or 0.32, would be heterozygous, and 4%, or 0.04, would be homozygous recessive. And we could do that with a Punnett square to kind of check our math there, and we would come out with the same percentage, the same relative percentage. So then we can kind of boil down the Hardy-Weinberg equilibrium to a calculation, to a formula. If P and Q represent the relative frequencies of the only 2 possible alleles in a population at a particular locus, then we have this formula right here. P squared plus 2PQ plus Q squared has to equal 1. So P squared would represent our homozygous dominant, Q squared would represent our, say, homozygous recessive, and then 2PQ would represent our heterozygous. Remember, we could get heterozygous in 2 possible combinations, PQ or QP. So where P squared and Q squared represent the frequencies of the homozygous genotypes, and 2PQ represents the frequency of the heterozygous genotype. So this is kind of our equation, our Hardy-Weinberg equation, or our Hardy-Weinberg equilibrium equation right here. P squared plus 2PQ plus Q squared is going to equal 1, or 100%. And that is probably more math than we've done in the last semester plus of biology. So this system operates kind of like a deck of cards. It doesn't matter how many times the deck is reshuffled. You can reshuffle that deck of cards over and over again and deal out new hands, but the deck never changes. If we're at equilibrium, at Hardy-Weinberg equilibrium, it's the same thing with the alleles of a population. Just because you shuffle the cards more and more often and deal out more hands and reshuffle, that doesn't mean that the aces grow more numerous than the jacks. You will always only have 4 aces and always only have the 4 jacks, and repeated shuffling of the population's gene pool does the same thing. It doesn't, in and of itself, change the frequency of one allele relative to another. So if you're at Hardy-Weinberg equilibrium, the frequency of any particular allele stays constant. All you're doing is reshuffling the cards and mixing those alleles around, but you're not really changing the relative frequency of any one allele. Again, recall that I said this Hardy-Weinberg theorem describes a hypothetical population that is not evolving, and you're rarely going to actually see that in nature. In real populations, allele and genotype frequencies do change over time, so we have to ask why, or what's causing that. Here the guys are. There's a Hardy right there and Weinberg right there. I always like to kind of show the actual people that are doing this work. I'm a big fan of science history, and I like to kind of read about the various scientists and people that were actually doing this work and passing on this information to us so that we could learn it today. And I think that's kind of cool to see photos of the various people in the past. So let's look at these 5 conditions that have to be met before you can say that a population is not evolving, that it is in Hardy-Weinberg equilibrium. And again, these are rarely met in nature. They would be, number 1, no mutations. So you can have no mutations occurring whatsoever, because that would instantly cause variation and changes in allele frequency. Number 2, random mating. No one individual can have any preference to some other individual mate. It has to be completely random. The combination of egg and sperm that come together have to be just a roll of the dice. Number 3, no natural selection. In other words, there's no gene or allele that gives any sort of inherent good trait or any sort of inherent advantage to any particular individual or allele because of the environment. So all the alleles have to be equal as far as natural selection is concerned. Number 4, extremely large population size. We're going to find when you have a small population size, you can have changes that occur quite a bit more rapidly, and it's not easy to control those changes and keep that equilibrium. So this really only works or occurs when you have a nice large population size. And then finally we have number 5, no gene flow. And gene flow, we're going to find out, is the movement in and out of a population of alleles that come from other populations. So this would have to kind of be an isolated population where there's no inflow or outflow, immigration in or out of other individuals. Natural populations can be evolving at some loci while being in Hardy-Weinberg equilibrium at other loci. So it's not an all or nothing. You can't just say, well, this population is not evolving at all. They may be evolving in certain genetic ways and maintain an equilibrium in others. So like I said, these are rare conditions, rarely met in nature, but it is a tool for comparing to the real world. I'll give you one example that is pretty good, a kind of a real world application applying the Hardy-Weinberg principle, and this is having to do with PKU. PKU is a genetic disorder. I'll try to pronounce it here. It's phenylketonuria. Phenylketonuria, PKU, is basically considered at Hardy-Weinberg equilibrium because, number one, the PKU gene mutation rate is low. It's not zero, but it's difficult to find zero, so we're going to do the best we can. The PKU gene mutation rate is fairly low, so it kind of satisfies number one. Mate selection is random with respect to whether or not an individual is a carrier for the PKU allele. This is a trait, a metabolic disorder in humans, and it results from being homozygous for a recessive allele. It occurs in about 1 in every 10,000 babies born in the U.S., and it's not something that will immediately lead to your death, but if left untreated, it can eventually result in mental disabilities and other physical problems. And nowadays in the United States, babies are routinely tested for this at birth, so you'll know whether or not you have this particular syndrome. And you can manage it. You'll notice that there are warnings on, say, like Diet Cokes that contain certain chemicals that can affect this PKU syndrome. So you can have a full life with proper diet and managing this particular syndrome. So at any rate, back to our Hardy-Weinberg. So the PKU gene mutation rate is fairly low. That satisfies number one. Mate selection is random. For the most part, when you meet a girl or a guy and fall in love, you don't ask them, hey, what's your PKU status? And you are or are not going to marry them and have children because of that. So that usually is not a factor. So we can say that mate selection is random as far as, in respect to this particular thing that we're discussing. Number three, natural selection can only act on rare homozygous individuals who do not follow dietary restrictions. So because we're humans and we're smart, we don't want to die, we test babies for this, and if the baby has this syndrome, we make sure that the parents follow a particular diet and ensure that that baby will have a long, healthy life. And that way we circumvent natural selection. We don't allow natural selection to do what it would normally do out in the wild. If you didn't follow the dietary restrictions, you would end up with problems and potentially dying before you reproduced. And why would you do that? So effectively, we're cutting out natural selection. That satisfies number three. Number four, the population is large. There's a lot of humans in the world, so that satisfies number four. And number five, migration has no effect as many other populations have similar allele frequencies. In other words, yes, we do have people from other parts of the world move in and out, and we have a mixing of populations, but because the frequency of this PKU allele is pretty much the same in all human populations, that really has no effect. So we can say that this PKU gene or allele is at Hardy-Weinberg equilibrium. So we can do a little bit of math. If we know that you have to be homozygous recessive to have this problem, and we know that the occurrence is about 1 per 10,000 births, that means the Q squared rate is 0.0001, 1 in 10,000. So our Q then, if we take the square root of that, is going to be 0.01. So if we know what the Q is, we can do some other calculations. We only have two possible alleles, P or Q, and if Q is 0.01 and we have to equal 1, well, you can do a quick subtraction, and that must mean that P is 0.99. So then we can finally do some other calculations, and we now know that P is 0.99 and we know that Q is 0.01. We can even calculate what percentage people are heterozygous. They would be carriers for this trait. That's 2PQ. So we can do that by 2 times our 0.99 and our 0.01, and that's 0.0198. And then we can even do a calculation to figure what percentage of the population is that. It's about 2%. So about 2% of the population is a carrier for this trait. They are heterozygous. They don't have the problem, but they have the possibility of passing it on to their children. So this is how the Hardy-Weinberg equations work, and we can calculate these various numbers and learn things about this particular population. We're going to pause right there and take a break. We'll come back and talk about the various things that disrupt Hardy-Weinberg equilibrium, because in nature you don't really see that too often. So let's talk about what we actually see in nature. That'll be concept 23.3. We'll take a break, come back in just a moment.